Okay, so this is the time for us to begin our session on protein misfolding and aggregation. So my name is Yury Chernoff, and I was asked to chair the session and give an overview and talk about some achievements and future directions in the field. So I would like to start from thanking the organizers of this excellent meeting for giving us an opportunity to exchange opinions on this and on the number of other important topics. And I hope that this session will provide a lot of mutually interesting information for all of us. So talking about protein misfolding and aggregation, and actually with the focus on therapeutic and diagnostic applications, first I would like to give a very brief overview of what is actually happening to the protein when it is synthesized in the in vitro system or in the cell. So when the protein is actually produced, the protein is initially unfolded, and then there are many different pathways the protein can undergo through. In fact, what is usually of interest for people producing protein for certain needs, or for the cell producing protein for fulfilling certain functions, is for the protein to achieve the native conformation. And then, if the protein is produced in vitro for the purpose of crystallizing the protein, then the native conformation can form a crystal, or sometimes for multi-subunit proteins it can form some fibers or other multi-subunit structures or oligomers. However, there are many other outcomes which are not as desirable for researcher or for the cell. And those other outcomes may constitute a significant problem both for the cell, because they're related to certain diseases, and for the researcher, because they prevent the researcher from producing functional or normally crystallized, normally structured protein. So there could be disordered aggregation of the protein, there could be hydrophobic collapse and formation of huge disordered aggregates. There could be formation of fibrils, amyloid fibrils, the ordered aggregates, which have the fiber-like shape. There could be also partial degradation, and those degraded fragments may again form the disordered aggregates, and they may form the amyloid fibers. So all those outcomes typically are not the desired outcomes. However, there are exceptions also, because in some cases amyloid fibers may also play biologically positive roles in the cells. In either case, it's very important to understand what actually influences this choice between the different phases for produced protein. And this is actually what the topic of our session is about. So, speaking about amyloids specifically, they are probably of most interest here because, in contrast to disordered agglomerates, they have the highly regular structure, and they can actually reproduce the structure. The structure is formed by interactions between certain regions forming the amyloid axis of the fiber. The rest of the protein is not necessarily involved in this axis. And then in this area where the subunits interact, they form a very strict interactive interface, very rigid interface, which is very difficult to break. So once the amyloid is formed, the amyloid is essentially almost unbreakable by regular means. And this interface is typically in most amyloids is formed by interactions between beta structures which belong to different molecules. So it's in most of the cases amyloids are cross beta fibers. So now the specific type of amyloids called prions were initially recognized by the ability to transmit the amyloid state. So if you actually take a prion, oligomer or polymer, and introduce this into the other organism or other cell in which there is no amyloid state for this protein, in which the same protein is present in the normal soluble state, then the prion would actually seed this formation of polymer and essentially turns the protein into the amyloid state. And in the process of this immobilization, the protein would change the conformation and becomes a unit of an amyloid. And as a result, actually, those prions become infectious. So initially, as I said, it was thought that there is a huge distinction, principal distinction between prions and other amyloids. But in reality, this process of nucleated polymerization in the solution, this process is characteristic for all amyloids. And by now, we know that many of the amyloids actually possess the prion-like features. And they actually are transmissible, are infectious, at least in the cell, at the cellular level. So there is really no such a strict distinction between prions and other amyloids. So that's more quantitative than qualitative difference. And I think Larry Walker will talk more about this later today. So if we talk about, first talk about protein aggregation in vitro. Aggregation of the proteins produced for certain purpose. For example, the protein and peptide-based drugs like antibodies, hormones, which actually are very widespread by now. So what is the problem with aggregation of those proteins? First of all, many of those proteins used for production of these protein-based drugs, in fact, have a high natural capacity of aggregation. Specifically, hormones frequently stored in the cells in the so-called secretory granules in reversible amyloid state. So if those proteins aggregate during preparation of recombinant proteins in Escherichia coli, they form inclusion bodies which are very difficult to solubilize and use, and very difficult or impossible to return this protein to the native state. So even if they isolated and prepared properly, they would frequently accumulate, aggregate with time, and also depending on conditions like low pH or high temperature may stimulate aggregation. Aggregate formation actually decreases drug efficiency, makes delivery more difficult, and also aggregate formation increases immunogenicity of the protein, because there are new epitopes formed. And for example, if it is a normal human protein, it would not be really allergic to humans, but if it forms new epitopes as a result of amyloid formation, it may become allergic. So as a result, it is a very significant problem, which essentially causes expiration of protein and peptide-based drugs. And it's actually quite difficult to understand what would promote this aggregation and what would prevent this aggregation, because our knowledge of mechanism is still not sufficient for prediction, at least not for every protein we know. Also, the problem is it's not easy to monitor formation of aggregates in the solutions of protein and peptide-based drugs, because existing procedures like size exclusion chromatography require significant amount of protein and are quite laborious. So it's not really that easy to do this in the small samples. So it's quite a significant problem in growing biopharmaceutical industry of peptide and protein-based drugs. So now if we come to the in vivo situation, then it's getting even worse, because for about 30 examples of diseases, so-called amyloid diseases, amyloids are actually causative agents leading to very significant health problems. The examples shown in red here, they will be mentioned either in my talk or later today, and you can see some very familiar names here, like Alzheimer's disease, Parkinson's disease, Huntington's disease, prion diseases, or transmissible spongiform encephalopathies, or glaucoma. So those diseases will be mentioned later today by me or by other speakers. I would like to point your attention to the disease like diabetes 2, type 2 diabetes, which actually was not considered to be amyloid disease until recently, but evidence accumulates that the cause of this disease is accumulation of a protein called amylin, or IAPP, in the amyloid form, which actually damages the work of the cells producing insulin, and this is why insulin is insufficient supply. And in fact, if you think about prion-like nature of those diseases, the sort of diabetes 2 to be infectious through blood transfusions is actually quite alarming. It has not yet been proven for humans, but there are some indications in the other systems that it might actually work that way. But let's go to those diseases which are better understood for now. So the Alzheimer's disease is probably the most popular one. And if you actually are lucky to live until the age of 85, then you have almost 50% chance of getting Alzheimer's disease. So its incidence of the Alzheimer's disease is actually increases with age. And once people get this disease, it's fatal and incurable. And that's actually quite a terrible death, maybe not so much for the person itself, himself or herself, but more for relatives, because that's actually associated with brain degeneration, and people actually losing the ability to control their actions, and the health costs are tremendous. You can see those data are a little bit outdated. It was like 3 years ago, was about 172 billion per year in United States alone, and I know it is growing. It was 7th cause of death. It's now 6th cause of death, and it's still growing. That's, if you look at the dynamics of death from different diseases like heart diseases, cancers, you will see that despite the large numbers of deaths from heart diseases and cancers, the dynamics is positive. There are less people dying of those diseases because some treatments have been proposed. There are more and more people dying of Alzheimer's because as far as people live longer, they're more likely to get this disease. So essentially, those are the most important diseases for coming generations, because with eradication of the other disorders, this is what is going to become #1 reason for people to die. So the other example is Huntington's disease, which is not as widespread. It is a heritable disease confined to certain families. It's called by mutation, by expansion of polyglutamine stretches in the protein called Huntingtin. And again, you have accumulation of aggregates in different areas of the cells, and you don't really know for sure which of those aggregated species is toxic and which is protective. There is a lot of work been done on this. So this disease is genetic disease. The other examples of well-known amyloid diseases are transmissible spongiform encephalopathies, and mad cow disease is probably the best known example. And let's see, before I go to mad cow, then also the other example, Parkinson's disease, which you can recognize here by image of Michael Fox, who is actually heading the foundation funding some of the research against Parkinson's disease. And he does have Parkinson's disease himself. Many other famous people actually acquire this disease. Muhammad Ali is actually another example. So this disease is not necessarily killing, but it really makes life miserable. And this disease actually also incurable in pure sense. You can only slow down progression. So now, the transmissible encephalopathies, the examples are Ship, Scrapie, chronic wasting disease, human Creutzfeldt-Jakob, and Kuru diseases. Those are actually diseases transmissible between organisms. This is an extreme form of amyloid-based prion. And you can actually get mad cow disease by eating meat of infected cows. So the frequency of transmission is not huge, but then the next problem is once it gets to humans, amyloid adapts, and so far we cannot tell what is going to happen at the next stage, at the level of transmission through blood transfusions. The long incubation periods for those diseases, frequently 10-15 years, make very difficult to make real predictions. And this disease actually led to extermination of potentially infected cows. About 1 million cows were killed in Europe, and massive beef recalls in the United States. Some of those were very recent. And in the United States, there are only a few cows, only a small portion, less than 1%, actually about 1 cow out of 1,000 has really been tested for this prion disease, because it's not considered to be a major danger here. But then In Canada, there were a number of cases, and I'm not entirely sure that US beef industry is correct in not considering this a major danger. So now, for all of those diseases, the pathway of disease begins from misfolded protein. Then it undergoes oligomerization, conformational conversion, forms the large amyloid deposits, and eventually it all leads to cell degeneration and death. Many of the diseases I mentioned, they cause degeneration of neurons. So because of that, it is a brain degeneration, which eventually causes the death of organism. So now, for genetic diseases like Huntington and some rare forms of Alzheimer's and Parkinson's, this is caused by mutation. For infectious diseases like prion diseases, this is caused by infection, getting the misfolded protein from outside. For sporadic diseases caused by spontaneous misfolding, spontaneous nucleation, which is very little understood. And in all of those cases, there are problems. Effects of mutations are poorly understood. Factors modulating misfolding and initial conversion are not known. And mechanisms of cytotoxicity are not clear. It's not even clear which kind of aggregated species is really toxic and which kind might be really cytoprotective. So the prophylactic measures as a result for most of those diseases are non-existent because it's not clear what causes the disease. And therapeutic treatments typically address the consequences rather than eliminating a cause. The other problem is because disease progression takes so long, it's very difficult to study this in the animal models. So it may take years. In humans, it may take sometimes many years until you get to the first symptoms. And that's certainly too late in most cases to treat the disease, and purely diagnostic approaches are almost unavailable. So now here I give some examples, not a complete list, some examples of treatments which are available for some of those diseases, and what actually is common among those treatments. All those drugs partly ameliorate the symptoms and slow progression of the disease, but they do not really target the actual misfolding. So the cause of the disease remains, and that means that earlier or later it will overtake the treatment. So then here, general summary. Most of those diseases are fatal and incurable. Existing drugs ameliorate symptoms rather than target the cause. Environmental factors have not been systematically studied. Prophylactic measures are essentially non-existent. And for most diseases, early detection assays are at rudimentary stage. It actually gives us some idea about the future, what we really need to focus on to get those diseases actually cured, eliminated before we all die of them. Because this is what is going to happen if we eliminate all other diseases. So now, at Georgia Tech, we have organized, actually Georgia Tech and Emory, we have organized the Center for Macromolecular Assembly Disorders, which, among other macromolecular assemblies, targets also amyloids, prions, and protein misfolding and aggregation. So this center includes 19 faculty members from several departments of Georgia Tech and Emory University. Here you can see the composition of the center, and many of the people who are members of the center are speaking at the conference, and some of them are speaking later today. So we have actually 15 faculty members from Georgia Tech and 4 faculty members from Emory. Not all of them are actually studying amyloids. We also address other macromolecular assemblies, but about half of the labs, at least to a certain extent, address amyloids and protein misfolding and aggregation. So the center actually organized, has established the seminar program with external and internal speakers. We did actually participate heavily in organization of SADET Symposium this year, which was focused on protein misfolding. Raquel has established the journal club in the center, which is also attended by members of Andy Bamerius' lab and my lab. And the integrative course on protein biology has been developed and is now taught at School of Biology, but we also welcome students from other departments. And this course is actually, we get the members of the center involved each year as guest speakers, providing guest lectures on different aspects of the field. We're now starting to develop the new graduate level seminar in protein science, which as an expectation will actually go even beyond the protein biology class. Initially would be attached to protein biology class, but the expectations are that we will develop this into an integrative, interdisciplinary course. And more than 10 collaborative research papers are published in recent years. We have the multiple PI grant, which is now active, and we're now thinking and discussing heavily the preparation of the team grant proposals coming from the center. So this is actually our plan of attack of the topic with the capabilities we have here at Georgia Tech and Emory. So the talks we will have today will be actually all presented by the representatives of the labs included in our center. So we will have Sven Behrens talking about approaches to assessment of aggregation, and Raquel Lieberman about the new glaucoma-associated aggregated protein myosin. Larry Walker will be talking about pathogenic protein seeds as a target for Alzheimer's disease, and Jonathan Rubin from Andy Bamerius' lab, and this work is also collaboration with Sven Behrens and my labs, will be talking about ion-specific effects on protein nucleation and strain formation. So this is the program of our today's meeting. Now I will try in 5 minutes to give a very brief outline of what we are doing in my lab that is related to the topics of today's talks. And of course I won't have time to go in detail. I will basically, instead of describing the topics of research, basically just give a very general impression of what they are about. So we're actually using yeast and in vitro models for studying amyloids and prions. Yeast is a unicellular eukaryotic organism, and you can see prions. Here there are 2 cells because it's actually dividing, and you can see prions in yeast actually by forming those fibrillar structures by fluorescence microscopy. And you can also detect them, what is very useful, detect them phenotypically by growth. No growth, growth, this is a prion. Red color, white color, this is a prion with white color. This is how easy you can detect prions in yeast. And actually those 2 strains, they're not different in anything else but prions. So they have identical genomes but different phenotypes, which actually tells you that it's not enough to know the genome composition, but you also need to know the proteome composition for deciphering what the organism makeup actually is, the heritable makeup of organism actually is. Those are heritable differences, those not just phenotypic differences, they will be passed in generations because prions would be infectious through cytoplasm. So one of the topics of research in our lab is the yeast model for Huntington disease. And here the huntingtin, human huntingtin, it should have extension in this polyglutamine region to be cause a disease, because this extension, if it's going over 40, leads to aggregation. So we actually reproduced a very severe form of Huntington disease in yeast, having the extension up to 103Q. So humans do not live long with this extension. Yeast can for a while, but they would eventually die as well. And we also found that if you put the proline region next to this extension, which is also present in Huntington, this proline region partly ameliorates toxicity because it actually directs aggregates to different targets, different locations in the cell. So most of this research is published, and the general conclusions are that endogenous yeast aggregates, called yeast prions, actually promote polyglutamine aggregation. Some aggregates are cytotoxic, others are cytoprotective. What I put here in red is what we did not know before the yeast model. Polyproline regions targets polyglutamines to cytoprotective aggregates. Sequestration of other proteins leads to cytotoxicity. Polyglutamines may cause toxicity by sequestering different proteins depending on what kind of endogenous aggregates you have in cells. That actually explains why in different cell types you may have different toxicity of one and the same type of aggregates, and people with different with the same length of expansion might actually develop disease with different onsets. So, and antagonists of polyglutamine toxicity can be actually screened in the yeast assay. So most of that is actually published in the papers coming out recently. So this other direction of our research, most of that is not yet published. We're preparing it now for publication. And yeah, I didn't mention that students from my lab, students and postdocs from my lab who participated in this research, Nina Romanova, He Gong, Kavita Gahalia, they now left. Gary Newnham, my lab manager, is still here, so he's helping us to continue. So now this research was done by Meng San, who also has left now, has graduated, and by Andrei Romaniuk, who is still at Georgia Tech, but not in my lab anymore, and also by Pavitra Chandler-Movlishwaran, who is actually continuing this research now in our lab. So this one is actually an application of yeast model to study Alzheimer's disease. And here we try to model how I-beta, the peptide associated with Alzheimer's disease, how I-beta can actually promote nucleation, de novo nucleation of aggregates. And here we fuse it to portion of the yeast prion protein. Yeast prion protein works as a reporter. And the most important thing here, you see this growth means aggregates are nucleated here. And if you actually put, and it's not nucleated in this system by sub-35 protein alone. And if you put here mutations in I-beta, which actually knock out formation of amyloid, you would not actually, that was done in vitro, that it was shown that they knock out amyloid, they won't form nucleation, they won't cause nucleation in yeast either. So there is a very good consistency in the system. So, and actually the same assay, we have shown it to work for PRP, alpha-synuclein involved in Parkinson's, and expanded polyglutamine involved in Huntington's. So it seems to be generally universal assay for majority of aggregated proteins. And the advantage of this assay is that in contrast to existing treatments, this assay actually can target the cause of the disease, formation, initial nucleation of oligomers. And because of that, we can actually use it to screen for agents and mutations, specifically targeting prion and amyloid nucleation. And so the final slide I want to show, this actually relates to our attempts to introduce new approaches for amyloid detection. Those can be used in solution for detection of amyloids and protein and peptide-based drugs. Those could be potentially also developed in early detection assay for some amyloid diseases. So this work was done again by Andrei Romaniuk and Meng San, and also with participation of Gary, who is continuing it now. And here I cannot really tell much about details of the assay because this all is not yet published. I can only show the outcome to prove that the assay works. And you can see here we have a mixture of two amyloid proteins in this case. And actually this mixture also includes a lot of other proteins. They're not the purified. They're in the rough extract here. Those two proteins are in amyloid form here. Here none of them is in amyloid form. Here only one, protein 2, and here only one, protein 1. And if we boil those proteins, yes, if they're pre-boiled, then amyloid form for those proteins is actually destroyed. So you can see that our assay clearly detects amyloids when they're present and when they're not boiled. And they're boiled, it doesn't detect them. This amyloid is not present, it is not detected, only this one is detected. So the protein is still there. It's only not in amyloid form. Here the proteins remain there, but they're not in amyloid form. They are not been detected. So this assay is actually very specific to amyloids. And that was initially funded by NSF, but NSF is not going to fund the applications of this assay, especially if they're health related. So we're actually looking for funds now to get applications of this work funded. So now I did not mention other people in my lab who are working on other amyloid projects. Katie is working on species barrier and amyloid poisoning. Dennis on cellular control of prion propagation. Beck on asymmetry of amyloids aggregation and cell divisions. And we collaborate with Keith Wilkinson lab on role of ubiquitin and cytoskeleton in amyloids. So that just give you a general impression. I cannot take more of your time, so we have to go to remaining talks. So let me see. I think we have 2 minutes for questions, and then we'll actually go through the next talk. So do we have any questions right now?
Can you elaborate a little bit? You mentioned the possibility that type 2 diabetes might be transmissible in blood transfusions. Is there evidence for that, or is that still speculative?
Well, what I know, and you probably know some of that as well, that this has been reported at the conferences, right, by Claudio Sotti lab. But I still did not see the paper published.
So. He's talked about A-beta, but not type 2 diabetes.
He also has data on amylin, yeah. So he was actually, maybe you did not go to that conference.
I think you're right.
Yeah, it was the next one, yeah. So, but he still did not publish it in formal way, so I can only refer to reports and more than one conference about this data. However, the involvement of amylin is actually well known, and I think more and more people accept this as a causative agent.
You showed something very interesting in the Huntington disease where the polyQ are aggregating, but they're providing cytoprotective effect by sequestering other proteins. Do you think this could be used something like cancer research, sequester maybe oncogene or something that would cause cancer and not take it out of the equation?
It's interesting thought. Potentially it could, especially if we can get sequestration by the non-toxic polyglutamine aggregates. And we do have some aggregates which are non-toxic but may still cause some sequestration. It's actually a good thought, but I don't have any evidence at this point that it would work for therapeutic purpose. But I think I don't see a reason why not. I think it's actually a good suggestion.
So can you differentiate between the two types?
Yeah, we can, but there is also some complication because this also depends on what kind of other aggregates, endogenous aggregates you have in the cell. So in the specific conformation, in the specific combination of other aggregates, you can differentiate between two types of those assemblies, and they have different locations in the cell, and they're associated with different structures within the cell. But this becomes more complicated if you move to a different cell type with different aggregation of endogenous approach.
Actually, what could happen, one scenario, is that they don't have to be cytoprotective. They just have to sequester a bad protein or oncogene, and then if the cells go into apoptosis...
Yeah, for cancer cells, again, you don't want to protect them. Yeah, so that's a good idea, actually. That's a good idea. Yeah, that's an interesting suggestion. Okay, so unfortunately we have to finish, unfortunately for me, but fortunately for other speakers, we have to finish with that, but I would be happy to discuss more after the session. And our next speaker is Sven Behrens from School of Chemical and Biomolecular Engineering here at Georgia Tech. Sven is going to talk about fast and non-invasive assessment of aggregation trends in protein solutions.